Have you ever wondered what makes you you? It's all in your DNA. Deoxyribonucleic acid, or DNA, is the instruction manual for life. This incredible molecule holds the genetic code that determines our traits, from eye color to our risk of certain diseases. DNA sequencing is like deciphering this code, revealing the precise order of the 4 chemical building blocks, or bases, that make up our DNA: adenine, thymine, guanine, and cytosine. Knowing the sequence of these bases is crucial for understanding how our genes work, how they differ between individuals, and how variations can lead to health problems. It's like understanding the letters and words in a book, only in this case the book is the blueprint of life itself. DNA sequencing has revolutionized many areas of biology and medicine. It allows us to diagnose genetic disorders, develop personalized therapies, and trace the evolution of life on Earth. By comparing DNA sequences, we can understand the relationships between different species and how they've changed over millions of years. One of the most significant breakthroughs in DNA sequencing came from a brilliant British biochemist named Frederick Sanger. In 1977, Sanger developed a groundbreaking technique that revolutionized the field, the chain termination method, now widely known as Sanger sequencing. This ingenious method earned him his second Nobel Prize in Chemistry in 1980, a testament to its profound impact on science. Sanger's method, elegant in its simplicity, relies on the basic principles of DNA replication, the process by which cells copy their DNA. He cleverly incorporated modified DNA bases called dideoxynucleotides, or chain terminators, into the replication process. These special bases halt DNA synthesis at specific points, creating DNA fragments of varying lengths, each ending with a labeled terminator base. Sanger sequencing involves 3 main steps: preparing the DNA, separating the fragments by size, and finally reading the sequence. First, the DNA segment to be sequenced is amplified, meaning many copies are made. This is done using a technique called polymerase chain reaction, or PCR, which we'll discuss later. During PCR, the chain terminators are incorporated, creating the DNA fragments of varying lengths. Next, these fragments are separated based on their size, using a technique called gel electrophoresis. It's like a race where the DNA fragments run through a gel, with the smaller fragments moving faster and further than the larger ones. Finally, the gel is read, revealing the order of the DNA bases. Each chain terminator is labeled with a different fluorescent dye, allowing us to identify the final base of each fragment. By reading the labels from the smallest to the largest fragment, we can deduce the original DNA sequence. Section 4, from gels to machines, automating the process. While manual Sanger sequencing using gels was groundbreaking, it was time-consuming and labor-intensive. Thankfully, technology has advanced, and we now have automated Sanger sequencers. These sophisticated machines streamline the process, allowing us to sequence DNA faster and more efficiently. Instead of using radioactive labels, automated sequencers use fluorescent labels, one for each base. The fragments are separated by size in a long, thin capillary tube, and a detector reads the fluorescent signal as the fragments pass by. This information is then fed into a computer, which generates a colorful electropherogram, a visual representation of the DNA sequence.
Section 5. Reading the Book of Life, Sanger sequencing in action. Imagine we want to sequence a short DNA strand. Let's say the original sequence is TATC. After the sequencing reactions, we'll have fragments ending with each of these bases, T, A, G, and C. These fragments are separated by size in the sequencer. The smallest fragment, representing the first base, will appear first. Let's say it's labeled with the color representing T. The next largest fragment, representing the second base, might be labeled with the color for A, and so on. By reading the colors of the peaks on the electropherogram, we can reconstruct the original sequence, tag C. Sanger sequencing, although not the fastest or cheapest sequencing method available today, remains a gold standard for its accuracy and reliability, especially for smaller scale projects. It's like having a trusted magnifying glass to examine the fine details of DNA, providing invaluable insights into the intricate workings of life.